Right? Synthesizing. Taking 2 units and building it. And a hydrolysis reaction is a decomposition reaction. See how we're starting to link these terms together, these concepts together. So, to build requires energy. To break down releases energy. Same idea. Okay? That's what we're talking about. When I synthesize, I need energy. When I break down, I release energy. Same idea. There's our water. Okay. All right. So then we talked about our monosaccharides. Those are the building blocks. Stopped over here at the lactose. We didn't worry about those other two just yet. Then we talked about the 3 disaccharides: sucrose, maltose, lactose. Those are the 3 disaccharides that are important for us. And we looked at polysaccharides. This one is what we store glucose as. We store glycogen in our liver and our muscles primarily. Starch, storage in plants. Cellulose, a building, or not a structural, rather, carbohydrate for plants. We cannot digest that. That's the takeaway for that. So then we started looking at lipids. What is the big concept about lipids and water? They don't mix. Okay, so when we think about these molecules, they mix with water. These do not. And that's going to be important as we move forward today or next week. Now, formation of a triglyceride, there are the building blocks, fatty acids, long chains of carbon, with a lot of hydrogen and very little oxygen. That's why they don't dissolve or mix with water. Take these 3 and you add them to a glycerol, and that forms triglyceride. 3 carbons, oxygens attached to the fatty acids. This is what you store fat cells, where you store energy. You break these down and get a lot of energy. Oh, I wish it were so easy. But that's your storage form, and there's the water. Okay? That's where we finished last time. Any questions? So, some examples just to show you. Now, if you were to read your food labels, you will likely see palmitic acid listed there. You will likely see stearic or linoleic. You'll definitely see linoleic on a lot of your food labels and other places. But those are incorporated in the foods. They're added for various reasons. Where do you think palmitic acid comes from? Not from your palm, but where does it come from? Palm trees, date palms, right? That's what's known to get this acid. And it's a saturated acid, and we incorporate it into a lot of things. Okay? It's kind of an emulsifier. It's something that we add to a lot of foods. I'm not going to ask you that. I'm just wondering if you look up there and saw that. Now notice these are saturated, so there are no double bonds. This one is an unsaturated acid. That's double bonds. And so this then is a triglyceride. Now, these lipids, there are others. There are others than just the triglycerides. The 2 others that are important for us are phospholipids and steroids. Phospholipids have a glycerol backbone. So that sounds like, or looks like a triglyceride. There's the glycerol backbone. It has fatty acids. But in this case, it only has 2, not 3. So what attaches to this 3rd carbon? What we call a phosphorus group. And there's usually something attached to that phosphate. And so this is not a triglyceride. We call it a phospholipid. Two fatty acids, 1, 2. Phosphate group, right there. Glycerol in the middle. Now, the big idea is that these are fatty acids. And what do we know about fatty acids in water? They don't mix. And so we call this the non-polar end. There are no charges down here. It doesn't have a charge to it. So we come on this side of the glycerol, and we see a phosphorus group. There's our phosphate. This is an ion. There's a negative charge. Okay? And come out here. There's a nitrogen. There's a positive charge. This is a charge group. Will this mix with water? It has charges. Yes. So this end is called polar because it mixes with water. So we would say this end is hydrophilic, mixes with water. This end is hydrophobic, and it avoids water. So that's going to be a huge idea on Monday, that there is this molecule called phospholipid, which has an end to it, which can mix with water, and an end which doesn't mix with water. We're going to need that to make the cell membrane. Right, so that's where we're going with this idea. Now, steroids are cholesterol-based. And this is a molecule of cholesterol. You mix and match and change it around a little bit, and you make testosterone and estrogens and progesterone and other steroids. Now, cholesterol, of course, for many of us, kind of we get that immediate, oh, cholesterol, bad. No, cholesterol required for you to do what you do. You have to have some in your diet. Just don't want too much. You can make cholesterol in your body, but you can't make enough of it for everything that you need. That's why it has to be part of the diet. The usual source for cholesterol, of course, is meats, red meats especially, but you can get some from plants. So I just had my blood checked up and found out my cholesterol level's just fine. So my triglycerides, yay, okay. So, cholesterol. We'll talk more about that about December when we start talking about hormones. Because steroids are one type of hormone. So there's another picture of our phospholipid. There's the fatty acids again, glycerol molecule, there's the phosphate group. Hydrophilic, the little ball, that's the way we usually draw it. And the fatty acids are these little squiggly lines. Hydrophilic, hydrophobic. Keep that in mind. We're going to revisit this on Monday. All right. So then we move on to proteins. Now, I'm not making light of this to say carbohydrates, lipids are relatively easy, but they are when you compare it to the proteins. Proteins are more complex. There's a lot more going on with a protein. Not just in terms of how they're made, or rather what makes them up, but also when we talk about function. It's easy to say carbohydrates. What do they do for the body? For the most part, we would say they are for energy. It's easy to say what lipids are for. For the most part, they're again for energy and for structure. When we get to proteins, we'll see they do a whole lot of things. So they get a little more complex. Now, we have been talking about polymers. Remember, multiple units, many units. So again, a protein is a polymer. What is the building block, the monomer, individual unit. It is called an amino acid. And to join amino acids, we make something called a peptide bond, which is another phrase or another way of talking about proteins, peptides. So if I add, or rather, when I look at amino acids, there are 20 naturally occurring amino acids. So that means throughout life, all of life, there's only 20 of these that are naturally occurring. Keyword natural because that means obviously we can make something in the lab that are different. Okay. 20 naturally occurring. Now, when we look at just 4 examples here, we see that they all have this exact same structure. We have carbon in the center, we have a nitrogen group up here, and we have an acid group down here. NH is amine. This acid, amino acid, where the name comes from. Now, the red is indicating that's different. There are 20 naturally occurring amino acids. There are 20 different groups that attach to that center carbon. 20 different groups. Now, that's relatively small. That's pretty big. So they come in different sizes. This one has a charge to it. This one has a charge to it. Why they're labeled as polar. These do not. They're labeled as non-polar. So some amino acids like to be around water. Others, nope, stay away. It's going to be important when we talk about protein structure and function. Mixture with water, not mixing with water. There are 20 of those. Now, there are 3 of you in here that are biology majors, and you're going to learn all of those 20 and draw them. Okay. The rest of you don't have to worry about it. Now, this side chain is called a functional group. I should have talked that up there in just a second. And that's what gives it the unique chemical property. That's why we're labeling these or putting these together as non-polar. These are polar. There's others that we call neutrals. We call some basic. We call some acidic. Don't worry about it. I'm just saying they each have unique chemical properties. That's the big take-home message. And there's 20 of them. Now, if I put together 2 amino acids, we call it a dipeptide. Put together 3, it's a tripeptide. Put together a few more, we call it an oligopeptide. Oligo just means many. Put together even more, it's poly, and that's even bigger, right? And then finally, when we get over 100, it's a protein. Now, if we get up above 20, like a polypeptide, do not make the assumption that if it's 20 amino, if it's got 20 amino acids in it, all 20 are different. No. Protein doesn't have to have every single kind of amino acid in it. It's random, more or less.